Dai, così avviamo. Ok? Così almeno gli rimetto la presentazione. Ok, la registrazione sta andando? Sì. Sì.
Ma che necessario di cui qualche sì, la seconda però. Per cercare quello che poi fare. Sì, si può, insomma. No, no, abbiamo previsto, no, non va avanti. Sì, non puoi tempo. Instead of programma quando lo chiamano le stelle io adesso presenti anche qualche però studio non posso permettere perché comunque sia nel senso per gli italiani c'è due che erano parte in lei più ce ne sono e che poi siamo ormai che parlano una francese uno turco ma parlano è preferibile io sono solo l'unico che parla in italiano però sto ignorando solo orario ci siamo ciao no non serve
Cosa c'è adesso? Va tutto bene? Devo stare qua o posso girare?
c'è telecamera
È qua. Per far sentire anche da casa, magari
Vabbè.
qui troppo. Per salutarmi rimango un po' velocia. Peccato che sono sempre che facciamo. Sapete di cosa?
Buon pomeriggio a tutti, ci siamo tutti? Possiamo chiudere la porta che da fastidio la lasciamo aperta? Aperta così circolerà e siamo tranquilli. Ho Michele Casini, alcuni di voi già lo conoscono perché ha fatto il supervisore ad altri nostri colleghi. C'è un'esperienza in Nord Europa che ce la racconterà oggi e frequenta qui il laboratorio di biologia marina e con i studenti e colleghi dell'Università di Bologna, quindi ha una presenza qui a Fano sia come docente dell'Università di Bologna sia come Fano Marine Center per quanto riguarda tutti i rapporti che ci abbiamo insieme sul dottorato. Gestirà lui la lezione, vi lascerà il compito con l'istituzione, come gli altri già sapete, per il 12 giugno e l'esperienza del Nord Europa per noi è un'esperienza nuova, quantomeno per alcuni di noi, e quindi ci farà sicuramente piacere sentire la sua lezione. Grazie Michele, buon lavoro.
Grazie.
Ok, dobbiamo passare all'inglese però, per capire siamo tutti italiani. E poi c'è qualcun altro che non è che non parla italiano, un francese, sì, c'è ce l'ho visto, quindi dobbiamo parlare inglese. But why are you so far away?
Sono fatti.
Okay, so good afternoon. We are already in the 4 hours again. 1 year time we are already done again. So we try not to be too long today. But what I would like, so I will present you some, a story about the Baltic Sea, about the Northern Europe. Because as Mauro said, I have worked for maybe 20 years in the North Sea, the Baltic Sea. So I was studying in the University of Bologna. I did natural sciences many years ago. Then I think 99, then I won scholarship and went abroad in Sweden. And I was supposed to stay there for a month. Then I stayed 20 years as a prayer. It was a bit longer. Then I came back here in 2019, less. I didn't believe I can go. But all my scientific career has been spent in Sweden. And that is 2019 was back at the University of Bologna. So I will tell you a story about what happened in the Baltic Sea in the last 25 years, more or less, 35 years. And how many of you study fisheries? PhD in fisheries or something related to fish biology or ecology. Okay. But of course, during the presentation, there will be some words that maybe are not so easy to understand. So if I forget to explain something, you just screen. Okay, you don't tell me what is not clear. Okay. And usually I tend to ask many questions to the public, okay, because otherwise they get bored, they're bored of listening, start to sleep. So it's better that we speak a bit, okay? So we interact. Okay, so I will talk about mainly population dynamics of commercial species, the Baltic Sea, about the biology, and a bit about management in management. Okay. So this is more or less the outline of today. So we start with the general concepts about the dynamics, and everybody is studying basic ecology, yes, university, so it should be very easy. Then we go directly in the Baltic Sea situation, and we learn about what happened in the last more than 30 years, about the biology of the fish populations, and about the hydrography also in that area, which changed a lot. And then I will present you a project that we started in the Baltic Sea many years ago. Then now we have some results. So I will explain you why we started that project and why the project can be used well in management of some fish populations. Then we talked a bit about ecosystem-based fisheries management, how you know what it is. And then we're talking then about some management option that during the last years we have been thinking about in order to try to manage in better way the fisheries for the Baltic Sea fish business. Okay. You know where it is, the Baltic Sea? You don't even know where it is. Where is the Baltic Sea?
Between Norway, Latvia,
Between, okay, Sweden maybe.
yes,
Sweden.
the stripe between the right.
The stripe between, okay, Sweden, Germany, the Baltic states. Okay, yes. Okay, so we start speaking a bit about ecosystem dynamics and population dynamics. So this is a slide that I usually show with the natural scientist lecture in Bologna. So everybody knows what top-down and bottom-up is in ecology. So if we say that the system is driven by bottom-up processes, what does it mean? Somebody can answer. Okay. On the primary producers, ends up with the
with what is
experiment.
so with what is what we eat what is under us.
Okay. But everything is driven by availability of praise, let's say, the resources. So if something changed at the bottom of the food chain, everything will change accordingly until reaching the top first. And bottom up is the top down. The opposite. What does it mean? So if we change top predators, so if the top predators is down. Everything like that.
Yeah, so top predator disappear.
Yeah.
Quickly at this point for the chain.
Okay, the top predators disappear, what happens to the prey or the predators? They will
Increase.
decrease. And the top predator below
It's
decrease. And the top level, and the top level even before below. Okay, so there is a negative relationship between trophic levels, right? One change, the other change in the opposite way. Okay, from the top to down. So this is a figure that is published in this paper. This is quite old paper, but I think it's very nice. So if you have the possibility, you can read it and talks a lot about this mechanism, bottom up, top down. And then we have also the wasp waste, it's called. So this, here you have the bottom up. So it's something that is changing at the bottom of the food chain. And then this change propagates up to the trophic chain. This is top down, so it's something starting from the top. And usually it's driven by maybe, in our case, overexploitation. So the fact that maybe we fish too much on some species. And what happens up there, you will cascade down until sometimes to the level of primary producers many times. And then maybe, not everybody knows the wasp waste process is something that is starting in the middle of the food chain. For example, the level of the pelagic fish, and then it propagates bottom up in the higher trophic levels and top down in the lower trophic levels. And an example can be, everybody maybe knows the situation of the anchovy of Chile, of Peru, that area. That is very productive area. There are big biomasses of anchovies, anchovies, and so on. And when this population changes because of climate change, for example, the chain propagates bottom up to the top predators and top down until the level of primary producers. And for example, driven by El Nino. You know, El Nino, this is all this fluctuation that have big effects on pelagic fish and on the ecosystem via this wasp waste process. Okay. I have been talked about this because we'll talk a lot about top down, bottom up, this kind of mechanism, so just to be on the same page with everybody. So this is the Baltic Sea. So you were right, so this is in Northern Europe. And one very particular things about the Baltic Sea that you see that it's quite difficult even to think that it's a sea. It seems like a lake, right? So actually the Baltic Sea is connected to the open sea, so to the North Atlantic. You have a stick, like, no, there's no stick. So you see here on this western part, the Baltic Sea, there are the Danish Strait, but there is also this bridge linking to even to that much where it is again, like there is a big bridge, right? And this is the only part where actually the Baltic Sea is connected to the Atlantic Sea. So this makes the Baltic Sea almost a lake, so the salinity is very low. Is very similar to a lake actually. And this makes the Baltic Sea very prone to change due to For example, change in temperature, change in salinity, change in currents. So everything change very quick in this area because it's disconnected from the ocean. Okay, there are two main characteristics of what we are interested in of the Baltic Sea is that the biodiversity is extremely low in the system. So there are many marine species, but since the salinity is very low, actually very few species have been adapted to this sea in the open area. And this makes the biodiversity, as I said, very low. So it means that when the biodiversity is very low, the interconnection between species are very strong. Okay, if something happens to one species, this will make change also to all the other species because the linkages between the species are very strong. And the other characteristic is very sensitive sea. And this is because the salinity is very low, okay, almost a lake, as we said. So all the marine species, what we call marine species, are at the limit of their distribution. So it's just enough small change, further change in salinity that everything will change in the system. Okay, so it's very easy to change the geography and the biology of this system because there is no buffer. Okay, there is no resilience, we can say. Okay, in the sea we have 4 different main predators, not only in the sea, but maybe also in the Adriatic Sea. We can think that is the same. So we have a very strong fishery, and since the biodiversity is very low, actually the main fisheries are on cod. You know what is cod? Cod is Atlantic. Then we have sprat, it's prato, that is what we have in the Adriatic Sea. We have also, for example, in the north. There is herring, that is another bloody fish, and then there are some flatfish.
Some freshwater fish, right? Like piles or thousands.
Yeah. So now we speak mainly about the open sea. But of course, along the coastal areas, there are a lot of freshwater fish. So pike, perch, sticklebacks also that are also linking the open sea and the coastal area. But here we cover mainly the marine species. But it's true that another interesting thing of the sea is that the marine species interact very much with freshwater species, because actually the salinity is very low. Then we have climate change, that is not also a surprise, but since actually we are talking about the closed sea, actually the climate change impacts very strongly here. We have big problems of eutrophication, so algal blooms, especially due to the fertilizers and other things that are coming from the land. They boost the primary production and they cause eutrophication. And then, at least in the past, the big problem was pollution. Now this problem actually has been decreased a lot, but it's still, of course, a problem. Okay, one of the main characteristics of the Baltic Sea is that very close to the sea floor, we don't have oxygen. So there are areas with very low level of oxygen. Here you see this black line that is like a section of the Baltic Sea, and here you see this section. So in the middle of the Baltic Sea, you see that can be very deep, can go down to 250, 300, 350 meters. And all this area that is being black and red are area without oxygen or with very low oxygen. Here you see the scale. So it goes from 0, so complete anoxia, to 2.5. And usually we consider very low oxygen level, maybe below 2.5, 3 milliliter per liter or something like that. Okay, so you see that there are big areas with very low oxygen. And of course, you can start to imagine what can be the consequences of this low oxygen on the fish, for example. What do you think they do, fish, when there is no oxygen? What can they do? Sorry?
Depends on the species. Generally, they move. If it's a carp, it just lays there.
Okay. So if you can move, you move. So either you die if you don't move, and it happens, or you move. But you move where? Sorry? Okay, in the Atlantic water, so we see that some of the fish actually, when they encounter an oxygen, they move up. So even if they are demersal species, so they like to be on the seafloor, actually they become pelagic. So there is lots of oxygen down there. Or what do they do? They move up, or they move in more coastal areas where there is more oxygen. So more coastal areas means where the depth is not so high. No? So either they move up or they move closer to the coastal areas. This is, you see, the time series of the areas without oxygen. So with the oxygen equal to 0, okay, up there. So you see that if we go back to the 70s, 80s, early 90s, was not so bad. And then from the middle of the 90s, these anoxic areas started to increase exponentially. Then they kept quite constant. And here you see the map of the situation. So when you see this black color, it means actually that there is no oxygen at all. Okay, so we have, when we go back to the 80s, 90s, this is the situation. So you could see anoxia only in the deepest areas. This is what we see more or less now. Okay, so there are big areas with no oxygen or with oxygen that is very low. Okay, this is, you can see the legend. So nowadays, more or less 30, 40, sometimes 50% of the sea floor is dead. There is no oxygen. So the fish, what they need to do, not only the fish, of course, they need to, either they die, we said, or they need to move somewhere else. Okay, yeah.
So these are not very big.
Okay, can you guess?
Rotation,
Sorry?
rotation.
Yeah. So it is a multiplication. So multiplication why?
The nitrates increase the concentration of nitrates, increase the microbial respiration. I don't know that.
Yeah, one by the plankton or the biomass die. What do they do? They sink to the seafloor, right? And to the seafloor, they need to be degraded by, for example, by bacteria. And bacteria degrade in the biological matters. What do they absorb oxygen from the water, so they take away the oxygen from the water. So certification is probably the main reason. Something more.
Water certification.
Yeah, YouTube.
Change.
So the temperature changes. So the water is more purified. So there is the less mixing. Is
it also because of the basin, the structure of the basin that you have water exchange? I'm asking that.
No, yes. So that is yes. So that is the third reason actually. You have seen that you can see here. You see that these are the connections to the North Sea, so to the ocean. So actually in the last 30, 40 years, the inflow of water from the Atlantic Ocean to the Baltic Sea has decreased a lot because of climate change. Climate change changing the pattern of circulation of the air and of the water. So the inflow of the water, which is full of rich of oxygen from the North Sea, has decreased a lot. So actually, this area became without influx of from the North Sea. So we have said eutrophication, stratification, and changing the patterns of the currents. They made this situation. Okay, another important thing connected to the eutrophication is the catchment area. So this is all the area from where you see all the rivers are flowing towards the Baltic Sea. So you see that there are European countries. We have Western countries, we have Eastern countries, we have Russia also inside. So also from the management point of view, it's an area that is quite difficult to handle. Okay. And during the last 30, 40 years, there have been a lot of laws in order to decrease the amount of nitrogen, phosphorus used, first of all, in agriculture. So actually, the amount of nutrients coming to the Baltic Sea has been reduced a lot during the last years, but still the amount that is still present in the Baltic didn't decrease a lot. Okay? Because the water in the sea is staying for 30, 35, 40 years. And all these nutrients are kept in the sediment on the seafloor. So actually they are not coming out of the system. They stay there. Once they come there, they stay there for, I would say, almost forever. Okay, we see later on this. And then we talked about the political situation, because there are 9 countries around the Baltic. Now there are 8 European countries, and there is Russia also. Russia is looking at the Baltic Sea in 2 places. So you see that the place close to St. Petersburg, okay, that is Russian, and then there is this small part of Russia also there. And of course, Russia. Okay, now we have even bigger problems with Russia, but also back in time, also Russia didn't follow the European rules. Okay, so when we go in the discussion about fisheries opportunity, fisheries management, there were European countries, and then there was Russia doing what they wanted, basically. Okay, they didn't follow the European rules, but their own rules. Okay, this is the situation anyway about the Baltic Sea. This is the very simplified food web. So we have said already that, so we have at the top of the food chain, we have the cod. The cod is the demersal fish, so the fish that like to be on the seafloor, which can reach a dimension of, in the Atlantic Sea, maybe also more than 1 meter, 1.5 meters maybe, so the fish can grow quite big. In the Baltic Sea, they don't grow so big. They start at around 1 meter, maybe, or they did before. And this is because of the lack of the low salinity, okay? So they don't allow the marine fish to grow. And then we have 2 pelagic fish, Sprattneri, and then we have the rest of the food chain. Here we concentrate mainly on this area, which we call the Central Baltic Sea, or Baltic proper. And then you see some numbers. These numbers are just statistical rectangles that we use in management, okay, in data collection and in management. But I don't think we will see anymore this number. Okay, so we talk about this. Mainly about these 3 fish, cod, Sprattneri, but we focus on cod, okay, because the big change report to cod. And cod is almost an iconic species, because it's one maybe of the most important emerging fish in the world, I would say. Maybe there is bluefin tuna, there are other fish, but the cod is one of the most important. And the cod also, remember when if we go back in time, in 400, 500 years ago, there were all these boats leaving from Spain, from Portugal, from France, going to fish in the middle of the Atlantic in search of who they want to fish cod, okay, which earlier was desiccated and then dried, and then with the possibility to refrigerate the fish. Of course, they started to, these big fleets of fishermen started to be out there for many, many months, and they were able to catch huge amount of this Baltic cod population. But in the Baltic Sea, as many other species, the cod the cod is at the edge of its distribution, okay, because of this very low salinity of the waters. Okay, so this is the time series of these three species in time in the Baltic Sea. So you see the cod is the red one. And here you see something called SSB. You know what it is, SSB? It means spawning of biomass. So the biomass of the fish that are reproducing. And so they are big enough, they're old enough, that are mature, so able to reproduce. So these are the time series of cod. You see that was relatively low in the 60s, 70s. Then you see this peak. Then it crashed and it stayed low since then. So we didn't observe any kind of recovery of this stock in this area. We see later on, we speak a lot about this. You see this time series of cod, and in 2012, 13, we see later on why. And then we have sprat, that is the yellow line, which is the main pelagic prey for cod. Yeah. So it seems that when cod disappeared, actually the main prey for cod started to increase, probably because there was not anymore much predation on the stock. That's why we were talking about top-down effect. So when cod disappeared, the pelagic fish started to increase. Okay. So this you see that there is a bit out of the time series. Actually, last day I tried to update it. So you see this the full time series until the last year, and you see that the cod didn't actually recover. It stays very low until the last year. The sprat kept a quite high level, even if it was oscillating, and herring also stayed quite low level. But we will focus on cod. We try to understand actually what happened with cod during the last 20, 25 years. Okay, so in the Baltic Sea there are two cod stocks. You know that the population, so the species is divided into many different stocks, from biological, but also from a management perspective. So it's dividing stocks. We can call them populations, even if stocks and populations are not the same thing exactly, but they are different stocks. So living in different areas, reproducing in different ways, exchanging DNA, having gene flow, and so on. In the Baltic Sea, there are two stocks. One that is called the Western Baltic cod, which is living mainly in the yellow area. And then there is the Eastern Baltic cod, that is everything else that is living from this island, the north. Okay. So here we focus on the Eastern Baltic cod. There you see the distribution of the species of the Atlantic cod. So you see that is going from eastern coast of the USA and Canada to Iceland, Greenland, the North Sea, the Barents Sea, and so on. Okay, that is there. Okay, so now we see some biological changes of this population in time. So the first change has been a contraction of the distribution of the stock. So when the stock was at high level, it was more or less distributed everywhere in the Baltic Sea. Then when the stock contracted in numbers or in biomass, actually all the individuals became concentrated in the southwest. And you can guess why. When the stock is low, the population concentrated in the southwest, in this case. Why do you forgive me?
The production rate.
Well, that's for sure, but why? Why they concentrate? Why back in time they were distributed almost everywhere in the Baltic?
Because of the changes.
Okay, can be because of the change in hypoxia, okay, but also because when the stock size, so the population size of cod, which is a marine species, goes down, of course, the individuals tend to concentrate in the best areas. Okay? And the best areas, of course, they are in southwest because from what we have said, there is higher salinity, for example, because it's closer to the North Sea. Okay, so they have better condition for reproduction, also better condition to grow because the salinity is higher. So actually, if they can decide where to be, they want to be on the southwest, okay, because the salinity is higher. But if the population is very big, usually the stock tend to expand, okay, to avoid the competition between individuals. Okay, so they expand the distribution.
I was wondering, do we if the cod that there is today is actually like the Baltic cod which are reproduced, or is it like North Sea cod who came to colonize? Because if it's closer to the strait, it might be closer solid.
Okay, yeah, we have studied about, we see later on also about tagging, also to see the movements of individuals. And no, there is no fish from the North Sea coming in the Baltic Sea. It would be a suicide to do that, of course. Because in the North Sea, they find a lot of oxygen, salty water, it's not so warm. Instead, in the Baltic Sea, you find all these bad things that make the population not in good state, actually. But we have anyway tagging data confirming this. So there is no fish actually coming from the North Sea to the Baltic Sea. There is no. Okay, so there's been a contraction in the distribution of this code. Other two biological changes have been one, a decline in the size at maturity. You know that but also the other organisms, they start to reproduce a certain size, a certain age, of course. And we see that in the 90s, you see that cod started to reproduce more or less 35-40 centimeters, so when it was already quite big. And then this size and maturity declined. And in the last years, it's down to 20 centimeters. So it means that fish growing already when it is 20 centimeters start to reproduce. And this usually, Michele, maybe you can tell also, this is a bad sign. When the size of maturity goes down, it can mean that there is a big attack of, for example, fisheries on the population.
It is because of selective predator? We don't.
We see later. It can be because of many things. Another big change has been increased in parasite infection. So if you go out in the Baltic Sea, sampling in these years, and you take the liver of the cod, it's full of nematodes, full of parasites. And these parasites, these nematodes, have as final host the seeds. In the Baltic Sea, there are quite many seeds also. So one hypothesis has been that actually the increase in the seed population in Baltic Sea in the last 20 years have caused this problem of increased parasite infection, infestation in the liver. Yeah.
So,
No, it's this one. See, I don't remember what it is. Osculatomy. I don't remember what it is. But they are very disgusting. So when you are out in the survey and you look at this fish and you open the fish, you make all the biological analysis, they are full of this snap. Okay. We come back to this. So this is another problem which has been caused, we think, by the big increase in the seal population, because the seal is the final host of this parasite. Other change has been the decrease in condition. You know what is condition? There are many different index conditions. But in this case, we mean the relation between weight and length of each fish. So how fat a fish is. Okay. So you measure the length, you measure the weight, you make the weight divided by the length, and you see this ratio, how much change in time. Okay. And it's indication of the fatness. okay, quality of the fish. And this is the black lines that you see. The red bars instead show the proportion of fish in low condition. So with a condition below 0.8, which many times is considered like a starving level. Okay. So fish with a condition below 0.8 are supposed to die very soon, okay? Because they don't have enough energy to survive. And you see that this proportion has increased in time. Nowadays, if we go out fishing with our survey, at least in some season, we see that up to 30, 40, 45% of all the fish that we catch have a condition below 0.8. So there are fish that are only bone. So they're called the ghost fish. So when you fish them, there is no flesh. It's just bone. Okay.
It is for the fourth quartile.
Fourth quarter, it means that is September to, no, October, November. Okay. Okay. Instead, Q1 is the first quarter of the year, so January, February, March. Okay. So depending on the season, we have different situation, okay? But back in time, we had the whole population constituted by these big, fat, nice fish. And nowadays, 50% of them are just skin. The ghost fish are. They are called, okay. Then we will try to understand why this is the situation. The last thing that we see, let's see, if you are tired of it, then we make a break. And let me just, no, it's not late. Is the disappearance of the big fish. So we are seeing that actually the fish have been become thinner and thinner, but they have become also shorter and shorter. So if we go back in time, we could find fish very close to 1 meter of length in the 90s, for example. Then after the 90s, things started to change. They became worse and worse. And nowadays, if you go out again with your boat, you see that the whole population is made up of fish around 25, 30 centimeters. So maybe the amount of fish didn't decline a lot, but they become shorter and shorter, smaller and smaller. And this, of course, is a big problem. Why you think for the population is a problem to have small fish instead of big? Because with the population with the same number of fish, what is the problem for the population where actually the size is they are all the fish of the same size, basically. What is the problem?
The competition is different.
The one is that they increase competition because being of the same size, probably they eat the same things, so there is increased competition. Yes.
Productive investment. Small fish, less eggs. Small fish, less eggs.
That's correct, yes. So the bigger fish that produce more eggs, not on the linear way, this one actually. So we want to have big fish in the population. And the other problem is that about the condition of the fatness. So that there is a very similar relationship between condition and fecundity, so the number of eggs that we can produce. So actually in management, usually what we want to have left in the population, we want big fish. And if we can also have fish, because they increase exponentially the fecundity, so the number of eggs that actually they can produce. Okay, this is another way to see this. So that is the same figure as before. This graph instead is showing the length of the 95 percentile, okay, higher percentile fish. And you see that back in time, the 95 percentile was around 60, 65 centimeters. And nowadays it's 35 centimeters. So it means that the biggest fish that you find is maximum 40 centimeters, 35. Back in time was almost 1 meter. And so the big fish have disappeared. And this is a photo that is a very famous photo. This is one all the polygon mine was a Finnish, that unfortunately, but he's a Finnish guy. And this is the situation, remember, around the 80s, when we had this peak in the population. Actually, in that period, we had a lot of fish, but also very few fish. And this is a real photo. So you see that there were fish almost 1 meter, more than 1 meter long. And of course now the situation is not this anymore. Okay, this is what we were telling before. Here you see the relationship between the length of the fish and the fecundity on the y-axis. So the fecundity, that is the, I don't remember, it is relative fecundity, that is a potential fecundity. Okay. So it means how many eggs, how many oocytes a filament can produce related to its length. Okay. So you see that when the fish increase in size, the fecundity also increases. Okay. But as we said, not in a linear way, but it's an exponential way. So we want where there's like quite big fish, long fish in the population. And then the 3 colors show the condition of this fish. So you see that at the same size, at the same length, if the condition is low, actually the fecundity can be much lower than the condition, than if the condition is high. Okay. So again, we have already said this 3 or 4 times. In the population, we want to have big fish, long fish, but also heavy, so fat fish. If we can do something to have fat fish in the population. From a management perspective, many times we can act on the length. So we can do, for example, can work with the selectivity of the gears in order to have big fish in the population. But also we would like to have fat fish because this increase of the fecundity, the potential fecundity, also very good.
What about the quality?
Usually also the quality is increasing with length and condition. So there is not, it's not good only because of the number of eggs that you can produce, but also the quality. So that is also true. And this was a work done by my old PhD student, Monica, that she has been working with the microtome, microtome in order to make histological analysis of the fecundity, so counting the oocyte inside the gonads of the cod, and she was showing this relationship between length and fecundity. Okay, so actually, when we have a stock constituted by small fish and thin fish, actually, we have problem for the fecundity. Fecundity, so for their reproduction. Okay. This means that they produce few offsprings. Then we have a problem for the fishery, if we have small fish. This is the old figure that will explain you why. So you see this, the percent on the y-axis of the binomial that is fishable, commercially fishable. If we go back in time, the fishermen would land fish only above 30 centimeters. Okay. Those were already quite big. So what did they do? If they were catching fish below 30 centimeters, they could make what was called discard. So they could throw them back to the sea, okay. Which of course was a waste, because once we are catching one cod, throw them back. So it's dead, so it's not that you save one animal just because you throw it back. Many times this fishing fleet, they fish for 3, 4, 5 years, hours. So you can imagine that the fish that has been in the net for 4 or 5 hours, even if you release it after catching, is that. So it's a waste to discard fish back to the sea. But back in time, until 2015, more or less, it was allowed to do this, to do discard. You were catching something that you didn't like, so you were throwing it back to the sea. So maybe it was too short, maybe it was too thin. So you are allowed to throw it back, hoping to catch something better for those. So here you see what is called fishable biomass. So the biomass of the fish goes above 38 centimeters. And you see that back in the 90s would be up to 80–90% of everything that was fish. And then in the last years, so these times years at least, in 2013-14, only 20% of what the fishermen were catching actually, they were able to keep. Okay? For the rest, they couldn't keep, they couldn't land, they couldn't sell. Okay? So you can imagine what was the problem for the fishermen. It was the fishermen couldn't fill their quotas, at least in the Northern Europe. The management is based on quotas. So the fishermen have a maximum amount of fish that they can fish every year for each stock. So since actually the amount of fish above 38 centimeters was little, they were not able to fill their quotas. Okay. And then they had catches that was difficult to sell, because even if they were about 38 centimeters, they were quite short, they were quite thin, so it was very difficult to sell. And then this increased the discards, so the problem of throwing them back when they were too small. Okay. From 2015, there is a discard ban. What it means? It means that everything that is caught by the fisherman needs to be landed. So even if the fisherman catch something that is too small, they cannot throw it back anymore. They need to bring to land. And this is counted against their quotas. So they try to avoid to catch these small fish because they cannot sell them, but they are counted on their quotas. So they want to avoid catching them. Okay. This green jump, and then we have the ecosystem perspective of having small fish. So some of us said already before that one of the fish are all of the same size, which is probably the setup. Okay. Instead, when in the same population we have small fish, intermediate fish, big fish, it's likely that they eat different things. Okay. And this is what these figures show. Here you have cod length on the X axis, the frequency on the Y. And you see that when the cod start to be big, what do? They shift from eating benthos to eat more pelagic fish. Okay. So it means that if we lose all the big cod in the population, the predation on the pelagic fish is reduced. Okay. So the pelagic fish tend to increase. Okay. And that's what we have seen before, that the sprat population, for example, has increased when the cod went down. And this is because not only cod decreased in abundance, but also because cod decreased in size. Okay. So they were not predating anymore on pelagic fish. Okay. That is what this figure shows. Okay. So what we have, this is what, so the last two slides, so and then we take a small break. So we talked before about top-down effect and trophic cascade. So when the cod disappears, the upper figure, so the upper panel, A, you see the time series of cod again in black. You see the time series of sprat in white. Then we see that the cod disappeared, the sprat increased because of predation released, probably. When the sprat increased, what happened to the plankton? At least monitoring showed that the plankton went down. And when the plankton went down, actually the phytoplankton went up. But of course, these are statistical relationships, so it doesn't mean that there is cause-effect relation. It's just statistics. But it seems that in the Baltic Sea, at least we have observed this, what is called trophic cascade, top-down effects from the top predators to the primary producers. And it's something that I've been, for example, working with for many years when I was working there. So it's something that is called trophic cascade. And that is the situation of the 80s. So we have a lot of cod. with a lot of cod and big cod, the spread population was kept low. The zooplankton population was abundant, and phytoplankton was not so. The phytoplankton blooms in summer, especially, were not so intense. Okay. Then something changed in the 90s. So we have had the big overexploitation of the cod stock. We have had hydrological changes, so worsening of the oxygen condition, if you remember, that the anoxic areas increased. Actually, the salinity went down even more. So the cod population decreased a lot. This released from predation the spread population, that is increased. The zooplankton decreased and the phytoplankton increased. So one of the conclusions of these studies, now I indicate on mine, but there are many more studies, showing that actually the phytoplankton blooms that we observed in the Baltic in the last 15, 20 years were due to eutrophication, mainly, but also to the change in the whole food chain, okay, especially due to the disappearance of the cod, okay, from the system. This we jump. So this is the photo of the phytoplankton bloom in the Baltic Sea in summer. And many years ago, what was it, in 2011, I think, yes, was also the European Parliament that was very much interested in this trophic cascade and the fact that the phytoplankton blooms could be facilitated by the disappearance of the top predators due to overfishing, for example. So they asked many of us to go there. So we went to Brussels to present all these studies, and they were, very much interested in understanding this interplay between nutrient emissions and overexploitation on the big fish. Okay, this is the last slide. Let me see. Yes. This is usually quite difficult for me to explain for others to understand. So we do it after the break. Okay, so we are a bit more fresh. Okay. So we take 5 minutes and then we come back. No. Ce l'abbiamo messo in focale, sì.
And not funny. And I get to grab it. You hear it, that Vuole piccole. Non si fa bene, però non dai, no, per me nessuno. No, non da fare. Ho capito. No, perché mia madre si può prendere di nuovo. Va bene. Ok, posso. Alla fine, quando è stato la tesi, io poi dopo sono iscritta, sono di particolari, esatto, esatto, e adesso in realtà è quindi vedono la tanto ho fatto che tanti toni di considerazione, si trova mangiare cosa mai un cibo ma proprio che squadra una continuazione. Sì, io direi la scuola di la compagnia per le rimaste e quindi speriamo però se poi anche abbiamo messo più di tanti non so perché essere cosa fa vicini così e un po' di piccoli che lasciamo quindi invece di essere prendo voglio capire che non si può io la rimane c'è anche prima da che per riempire poi non mi grafici ma io ho fatto un altro verso il verso fanno poi che non si fa cioè quindi faccio una generica però non si dice dai studio del tanto delle sono bene, lo fanno se non sono molta la vita è 5 anni. Quindi in realtà non so che non ne ho pasti e ancora più complicati negli stati e quindi bisogna capire se riusciamo con io però non si guardi la questa primavera adesso poi passo per la vita però io e anche abbiamo studiato di faccio di casa e comunque ancora decorazionale deve essere che c'è un sacco e anche lì volendo c'è in qualche questa di un po' un jeans però non viene ora ma non è troppo sulla chiesa non ora una culture di che sono usciti avvistamenti poco hanno anche una beata, non so se le sapere vinto fare interessanti io anche se un po' voglio dire anche da quando insieme sono problemi anche invece andare a dire che mi vanno. Quindi sembra che alla fine l'infection sia stata praticamente calcolata e quindi siamo interpellati. Beh, e questo prevenire che può portare a una certa una prevalenza. Sono state esatto, prima cioè che non avete fanno prima, quindi mi volevo sapere, non era una cosa che non ci sono due, non vedo che c'era applicato una dopo, no, quello che con la quindi in realtà a livello di onda che da dopo voglio arrivare in qualcosa le persone a fare un esatto, quindi da fare un cappello nelle riunioni da un po' diverse però da poi la parola inizia è il pausa, io capisco l'idea da io credo che non mi sembra non ha bene non più non mi è la cosa sono che non è per la persona che non viene essere ho fatto da 3 mesi un gruppo riesci a trovare una migliore fare l'interesse. Non mi dico e poi io ce l'avevo in mano perché secondo me è una fare questo. Ed era un po' che non cominciava, ma è un quindi è bello insomma che fare qualcosa di lui. Io sono così e sì, però no. Mi pensiero che la messo e mi spazio in tutto c'è favore guarda non dice che abbiamo fatto delle gare ma qualcuno fa se vuoi dire come scrivere
Now,
Se no,
quando sale a fare treno, lenta. Vabbè, sapendolo avremmo fatto meno ore, siamo fermati di meno. Però secondo me, allora, sono le 4 dopo. Secondo me le 6 è finito. Sì, l'avremmo finito alle 6.
vabbè.
Sì, però io credo che intorno alle 6. Sì.
No.
Come si fa qua a togliere la doppia slide successiva?
Giusto, da qua perché in modalità elevatorio
No.
hai presente
Vabbè,
caffè e telefono.
non fa sempre. Ok, possiamo iniziare. Okay, so we can start again. So we finished this slide. Then if you have question, we can take it later. But let's finish this slide. So this is a paper published many years ago already. So it's 20 years already. This is Frank is Kenneth is called. He's a Canadian guy working that. And many years ago, so in 2007, did this study. And also this is very interesting. So if you are interested in this top-down, bottom-up dynamics, I suggest also you to read this. So this sometimes is complicated to explain because it's quite complex. But okay, you see the map up there, and you see different communities, different areas, different communities, where this Frank and colleagues took the data that have been already published in literature the year before. So all these dots, green and reds, are communities on which there are a lot of data about top predators, prey, plankton, phytoplankton, and so on. So there were time series in time. So actually, these authors, they wanted to see if these communities were regulated by top-down effects or bottom-up effects. And they want also to try to answer the questions why they were regulated by top-down versus bottom-up. So each dot corresponds to a community. The dots are the same also here in these plots. Each dot is one community that you see up there. And this plot is showing the relationship between species richness, so an index of biodiversity, whatever, and the correlation coefficient R between trophic levels. So it means that this correlation coefficient is positive. It means that it is an indication of top-down or bottom-up. Right? Instead, if the coefficient is negative, it's an indication of top-down. Okay? When one trophic level increases, the one below increase. Okay, so you have a negative sign of the correlation coefficient. And of course, if the sign is stronger, so -6, -0.6, -0.7, and so on, the top-down effect is stronger. Instead, when the correlation coefficient is around 0, it means that it's quite weak. Okay. So they plotted the species richness in each of these points with the correlation coefficient that they were found in literature driving these trophic levels. Okay. So if we look at this plot, what do you see? Can you give an interpretation to this plot?
The species is exploration is inverse. So when there is less species richness, it's more top-down driven.
Yes. And instead, when species richness is high, the communities are mainly driven by bottom-up effects, because the correlation coefficient is positive. Do you agree about this?
Also with them.
Third? Then we go there. Then let's focus on this. So according to them, they are saying that when we are in communities that have low biodiversity, it's easier to find top-down effects. Instead, when we are in community with high species richness, it's easier to see bottom-up effects. Okay. Can you try to explain why?
I think because it's linked to the complexity of the ecosystem. The more the complexity, it's easy to understand the example of the code. In Baltic Sea, we have less species, less diversity, top-down regulation. But why? Because we have a stronger relationships between species.
Like could be also bottom-up.
Maybe it needs more praise and less predators that are able to.
Much of this is related to the fact that there is not so much inter-specific competition. So competition between species. For example, if we take cod in the Baltic Sea, we said that there is a top predator, right, of the system. If the cod disappears from the system,
and we didn't sleep enough to go on this person, but if the code is appeared, there is another predator that can take the functional role of the code. There is salmon, and obviously there is quite a lot of salmon valuable abundance compared to the other species. So actually, the code disappears. There is no other species that means that it's much easier to have this problem escape.
Okay. Instead, if we were in a higher biodiversity system, if cod disappeared, there would be another species, maybe competing with cod, that would increase and would keep the role that cod had before. Okay. So the role as predator, right, of pelagic fish. Should double. Okay, that's why it's easier to see top-down effect than low biodiversity system. So actually, the Baltic Sea was not included in this study. So now we put it on the map. Where do you think we put the Baltic Sea in this panel? The dot of the Baltic Sea. Where do we put it? Bottom left. So somewhere here. So we have spread the top down. This, yes, yeah, all like that. So the biodiversity of the Baltic Sea is quite low. Now I try to guess a bit how many species there are in the Baltic Sea. From the study that I did, more or less, the coefficient of correlation is around 0.6 between trophic levels. So the Baltic Sea fits to this expectation, at least now. Then you were talking about temperature. So actually, they try to understand why, also what it meant, when there was more species richness in terms of temperature. So they show also a positive correlation between temperature and correlation coefficient, because many times higher temperature also mean higher biodiversity. Okay. So this is the first part that I wanted. Really. So actually the main point that we have seen the change was the ecosystem. So oxygen maybe. We're seeing what happened with the cod. So the cod abundance went down, the limit size of cod went down, the condition went down, the site maturity went down, the farmer site infection went up. So we'll begin with that. We have questions about this. Oh, okay. So now, in the second part, we try to understand mainly this figure. So why actually big cod had disappeared from the system? They said that back in time we had very big animals, 1 meter more or less. Now everything that we find is around 30 centimeters. Maybe the fisherman can find bigger fish, but when we go out with our scientific boats, we don't find anything bigger than 35 centimeters. Okay, everything is around 25, 30. So actually, the big question that we try to answer is why? Why big fish has disappeared? And again, to you, why do you think fish can disappear? In general, we can black. Vision, okay? In what way? Yeah, so the fishery took quite the time went down. Then it was the fisherman on get the big one, if I have done.
The fisheries
capture the code of GFT55 and the animals that can grow.
Okay. Okay, so you mean that the fisherman catch the cod before the
Yeah.
to grow? Okay, yes, okay, can be. So increase the mortality that, okay, the big fish are not allowed to live here. Okay, you get a lot of small fish, okay? Could be. Something more?
We got fish raising the fish of the nest. If I have a big fish, it's easier to catch it rather than a move one. Just a guess.
Okay, so you say that maybe the fishing effort has increased as soon as the fisherman got the big fish, they take the big fish out of the population. So
Maybe the spawning, where do they spawn? Where do
in the
they,
pelagic water, they have pelagic waters.
in the deep water or?
No, pelagic, so far,
Okay.
in the mid water.
So maybe if they fish during the spawning season, they catch the big ones. Because
So
they're.
you say that, okay, so the fishing effort has increased, but in the fishing of the big ones, they take away the big ones of the population. Instead, you are saying that they take away them before they have the chance to grow big, okay? Everything related to fisheries, that's something different.
Maybe, maybe the increase of the sea temperature, so if you, the temperature increase, the size of the fish decrease.
Usually, yeah. We didn't speak too much about temperature, but temperature increase mostly there, everywhere else. And one, yes.
You know, issues, they select, there's a selection on, the portion of the population that matures earlier, since premature or older, you have a lot of chance to transmit your genetics before being finished. So, and then once you mature, you use your energy to make eggs more than morning.
So still related to fisheries, yes, but there are different mechanisms that work about. Temperature, something like that. Talk about oxygen. The oxygen quality is important for.
Trophic pressure in general.
Sorry.
Trophic pressure in general, how the fish able to
Okay.
livestock or agriculture.
So not the pollution or propagation. Okay. So we've been speaking about selective fishing. So it means that that we take away the big fish from the population. Because, for example, the fishermen want the big fish, usually, no, right? So they take away the big fish from the population, and automatically the mean size increases. Okay. So it can be a selective fishing or selective predation by some predators. We were talking about seals before, for example. So seals can predate also on cod. So it can be size selective predation of seals on cod. Or there is something happening to the growth. So that the individual don't grow quick enough as before. It can be temperature, can be oxygen, can be other things. Okay, so let's see. Here I wrote high size selective fishing pressure. You told this. High natural mortality can be predation by seals, or can be due to decreasing growth. So they are still old, maybe, but they don't grow quick enough. Okay, so they stay small. And if it's due to a declining growth, actually we want to know when in the lifetime they start to grow slowly and why. And then from a management point of view, we want to understand if we can do something about this. Because actually, since the cod is the main important fish species in the Baltic Sea, the fishermen, the politicians, the ministries, they want to have the population of the cod back. Okay, so they want to increase the madness of the cod. They want to have back the big fish. So the big question for the managers is, can we do something about that, or is climate change? We cannot do anything about that, so we need to eat something else. So adapt to catch and eat something else. Okay, so we go back to this slide. So the question is this: is it due to low growth or increased mortality before the fish has had the possibility to grow big? Okay. So some of you have been working with the fisheries, age determination, yes. Usually to try to answer this way. Actually, for example, if we want to answer the question, the growth rate has changed in time or not. What should we do? Everybody can answer.
You can check the otics to determine the age of the fish and compare to the data set.
So we can calculate fish per. Then compare old fish to the new fish and see if the growth rate has changed. Okay. So usually we should do that. Okay, we take the otoliths. Otoliths, you know what they are, right? Otoliths. We age the fish and we see if the growth rates have changed. So if the size of the specific age has changed in time or not. So this is the growth curve of one hypothetical fish. So you have age on the x, you have mean length on the y-axis, and usually not only fish but many other organisms they show this growth curve, no? This growth curve. And then you can model with the different kinds of equation. Well, so you know that back in time, the fish could reach the size of, we said, around 1 meter, 90 centimeter, 1 meter. If we go back here, see that we observe some fish up to 87, 88, 90 centimeter. So that's what this line shows. At maximum, we have fish around 90 centimeter, okay? And around the 90 centimeter, they are 10, 11 years old. So if we had the otoliths and if we could age the fish, we would estimate this growth and understand if nowadays we don't have any more the old fish. So if we don't have any more the old fish, actually, by doing age determination, we wouldn't find old fish, right? So the growth curve would stay more or less the same, but we wouldn't find the old fish, okay? In this case, it would mean that we would have lost the big ones. So it's not the problem of growth rate, but it's a problem of losing the big fish because maybe the fishermen are taking them, okay? Or the second hypothesis is that actually the old fish are still there, but they're not growing anymore. So the maximum size at the same age 11 from 90 now is 50, for example, 60. So it means that there's been a decline in the growth rates. So the old fish are still there. So it's not a problem of selective fishing. It's a problem that they don't grow anymore, the individuals. And then there are all the alternatives between, okay? So now let's see if there is this animation, okay. So usually what we do to answer this question, we look at the otoliths, right? Everybody knows what otoliths are. Okay, so we don't need to explain. So these are the otoliths of what we call that. And if we go back in time in the 80s, 90s, this is what we call otoliths, of course, enlarged. And you see that we were able to see these bands, okay, that we count to estimate the age of the fish. But something happened after the 90s, so 95, 2000, 2005. You see that these bands that we used to estimate age disappeared completely from the otoliths. So actually, in the last 20, 25 years, we were unable to age the cod. Also this, we don't know exactly why these bands disappeared from the otoliths. but they disappeared. So actually, we were not able to answer that question. If it's a problem of growth, or if it's a problem of increasing mortality of the big fish, because we were not able anymore to age the fish.
They keep growing instead of present period.
They continue to grow.
Yeah, like
the operates.
slowly instead of grow and then stop and grow.
The operates.
Yes. So
Yeah, the operates continue to grow, but they don't show anymore
yes.
the annual increments, the bands that we used to age them.
And through the scales, it's the same.
Yeah, for cod, scales are not helping.
No.
Scales can be used mainly for eels, for salmon, for cod, it's not so valuable. So that we were not able to age the fish anymore. Is still there. And this was a big problem for management, because when we want to manage the impact on the fishermen on the population, since we were not able to tell that it is declining in size, growth, or mortality, actually, anymore to evaluate the status of the stock. Every year, there are international organizations that are evaluating the status of the different stocks around the world. And what is called for assessment, on what it is. But since these questions, then we were not able anymore to do stock assessment, so to evaluate the status of the stock. And the status of the stock is something that we need to have in order to provide advice to the managers. I try to simplify, tell me if you don't understand. So the management was complicated, and the MSC, that now I explain what it is, was suspended. You know, MSC and SP means Marine Stewardship Council. That is like the quality mark of the product. So the fleets, the fishing fleets usually apply to get this quality mark that indicate fisheries that is done in a sustainable way. Okay. And the customers nowadays, when they go to also yourself, I mean, when you go to the supermarket, for example, buy products like fish, maybe you can have this quality mark. I don't know if you have seen this mark, I don't know, this mark in the fish products. Okay, now I should, I'm not supposed to show any kind of, to make any kind of a reclam of this, but things very good. But if you go to the supermarket, you see it's quality marks. Some have quality mark, some don't have. And the fishermen, the different fishing fleets in the different areas where the different stocks, so fishing different stocks, apply to have this quality mark, so they know that the customers are looking a lot about this. So it's easier to sell the products if they have this quality mark. So since we were not able to do stock assessments, so the evaluation of the stock status from 2019, this quality mark, which the fishing fleet in the Baltic Sea had on cod, lost this quality mark. So the fishermen didn't have this quality mark anymore. And this was a big problem for them. Okay. So the big problems were that we were not able to evaluate the status of the stock. So if it was in good or bad status, we were not able to give scientific advice to the managers. The fishermen were not able to remember to catch their quota. They increased the discards. They lost this quality mark. Okay, so it was a huge problem, the Baltic Sea. And this because, again, we couldn't answer the question, is problem of growth or problem of mortality? And so we needed to answer that question somehow. So what we did was to start a tagging project. Tagging project was in that we were catching fish, we were applying tag to the fish, so in this case of the dorsal tag, that we will see later on. So here, see, so we were fishing individuals. We were applying a tag. We were using this spaghetti tag, they are called because they look like spaghetti, on the close to the dorsal fin. When they were caught, actually they were measured in length. Okay, they were applied this tag. They were released again to the sea using this cage, so that the fish were put down the same depth where they were caught. They were released, and then we hoped that the fishermen would catch them back on time. It's after some months, after some years, and so on. And so when the fishermen were catching them back, what do you think we did? We were there, we measured again the length. So from the difference between the release length and the recapture length, we were able to measure the growth in centimeters in that period of time. Okay. And so we were able to estimate the growth of each single individual. And we were lucky for this kind of experiment. They were done in the Baltic Sea, back in time in the 40s and 50s and the 60s. So we were able to compare the whole the growth rates of the stagnant experiment and the new growth rate for the stagnant experiment. So to compare the growth rate that changed in time. Okay. So there were four countries involved. So you see in black is German, in green is Denmark, in red Sweden, and in blue Poland. Okay. So there were these four countries that were covering the main distribution area of the cod. So each country went out with the boats, fishing, measuring, tagging, and releasing back to the sea. Okay. If you're interested, there is a link here to that with some results and stuff. So this is more or less, this is spaghetti tag, the yellow one. It's a plastic. You have it like a sounds so automatic, but like the one that you use to insert a stack close to the dorsal fin for the fish. That is a standardized methodology that has been applied for decades. And this, you see that on the spaghetti tag, there is some information. So of course there is a number of the fish, because when you capture the fish, you want to match that fish with exactly the initial length. There is a number of the fish. Then there is a telephone number. So the fishermen, when they call the one fish with the tank, they need to go to the Casini. So there was a telephone number. There was a website. So they knew that they needed to call us. Okay. And then, of course, we needed to. This is again, some small fish with the tag. Okay. So actually, what we needed was to make the fishermen aware for this project, because we want all the fish that the fishermen pull with tag. We want that tag measure different parameters and so on. So we were producing these. These are postcards, 26 meters by 10 maybe. This is in Swedish, but then there are also in Polish, in German, in all the different languages around the Baltic Sea, which are them. So the Latvian, Lithuanian, Estonian, Russian, so a lot of postcards with different languages. And on this postcard, there were very brief information about the project, why we did it, telephone number, contact person, and so on, because we wanted all the fish that they were catching, we wanted them alive. Okay. So these were postcards that they were sending to the fishermen, for example, for Christmas, present, we were sending this postcard. We remember them 2 or 3 times a year that this project was important, especially for them. Because you remember that they got suspended the quality mark and everything. So it was also important for them. So we tagged in 3 years, I think we tagged around 30,000 fish. And how many do you think we got back on the vision? 1,000. 15%. So 100 fish. 50%. So 1,000, 50%. 100%. We got 350. 10, some years ago. Now I lost a bit because then I left, but 1%. 30%, yeah, 1%. Well, that's 1%. And one of the problems was that there are different problems. One, that maybe the fisherman was didn't report what they did. Because sometimes the relationship between scientist and fisherman are not so smooth, not so nice, not so easy. So probably we didn't get back with fish with the right catching. But the main problem is that the fishermen now have on the boats what is called the gutting machine. So there is a machine that is cleaning the fish automatically. So they don't touch the fish. So all the catch is going in one machine. The machine is cleaning the fish. So actually the fishermen don't touch the fish, so they cannot see the back. That there has been a big problem, I think, in front. But we got back 350, 360. Okay. So the results. So we were able to compare the growth rates of these 350 fish with the growth rate of the whole diving experiment that I told you that we have in the road is 57, especially from Poland. Poland always done this big project forever. I don't know why, but they had a lot of data. So this is an example of the data that we got back with the tagging experiment. So each black line, what do you think is a single fish? Is a single fish. So there is a growth of each single fish. So the change in size, in length of each single fish. You see something strange with this? They lost weight. They lost weight. The problem is that it's not weight. It's length. That is the problem. If it was weight, it was not big. Yes, bigger. But the problem is that it's length. So what do you think? What's wrong? So when you see black line going down, there's something wrong. So he cannot use line. So what do you think happened? Because only half the fish. Sorry? Because only half the fish. I mean, probably that sometimes we got the measurement from the fish, right? Maybe it was not so much comparable with our own measurement. But just but some are, yeah, different. So of course here you have a different kind of measurements. You have mistakes. Also, many times that they were writing things by hand, also writing it's like wrong, or maybe the hand style is not so easy to understand. So there are a lot of these problems, of course, in this big experiment. Seems that there is a model. Sorry? There is a model. There is, okay. And then from all these kind of lines, you can fit the growth curve, okay, which is the red one. So by comparing all of this growth curve back in time from the 50, 60, 70, we could understand if there's been a change in this growth rate or not. So this is the result. So again, it is the same PhD student course that looked at the cavity before. So here you see a figure showing the growth rate, so in centimeter per year, of a fish that initially, so when it was caught and released, long. Okay. So if we go back in the 60s, 70s, a fish 40 centimeter long was growing around 7.5 centimeter a year. Then this growth rate increased to more or less 10, 11. And then in the last years went down to 4 or 5. So it means that at least compared to the 1890s, there's been a huge decline in the growth rate. So back in time, when we were growing less than meter per year, now they're growing 5 meter per year at the size of 40 centimeters. Okay. So it seems that there's been a decline in growth. So they're not growing as before. These are the different lines in different periods. So you see that from the 55-70, we had the highest growth rate, and the lowest is the last period, 16-19. So this demonstrates that the growth rate at least has gone down quite a lot. We didn't catch so many other fish in 2019 because the codfish has been closed in 2019 because the situation was flat. And the industries are closed to the fish. Fishing blackfish, processed by catch of fish, they take to some point. But the direct fisheries on cod is slow because it's so you don't have any fish to this actually.
Have been done some progress, years.
Yes.
Is there a progress?
So now with this new data about growth rates, we apply stock assessment, estimation, evaluation, and so on, and we were able to do stock assessment again. And it showed that the situation of the status is very bad. That's why we the decision is to close. But even though the fisheries have been closed for 6-7 years now, no recovery is so the stock is at the lowest level ever. So, okay, so the growth rate has gone down. Then other things that was done in this project was to tag not only the fish but also the otoliths. So by using is a specific antibiotics that is called tetracycline. So if by injecting these antibiotics in the fish, it creates a band on the otoliths, a fluorescent band on the otoliths. So actually, when the fishermen were catching back this fish and providing this fish back to us, we were able, by looking at the otoliths, to see what happened from the time we were tagging the fish, that is this yellow fluorescent band, to the time of the death. Okay, to understand more why we were not able to see any more this growth band of the otoliths. Okay, so it's a way that we try to use to understand why actually the otoliths didn't show any band anymore. And then since then, what we did, since we were not able anymore to wait fish by looking simply at the rings, we were trying different methods, like microchemistry, for example. So we were collaborating with Cornell University, for example, in USA, where they use this synchrotron. This synchrotron is a machine that is accelerating particles by close to the light speed. So they accelerate particles very, very quickly. And so what they do is they use this accelerator to tweak the otoliths with the X-rays. And the otoliths emit, so the electrons in the atoms of the otoliths emit other X-rays that you can use to study different elements within the otoliths. So you can see, for example, strontium, barium, and other kind of elements that can be used to age the otoliths. I think there is one synchrotron also in Switzerland, I think under the one. This one synchrotron is accelerating particles close to the light speed. So that is another technique that you can use to try to see if you have seasonal bands of different micro elements in the otoliths. Another way is start to use laser. So there is a laser that is hitting the otoliths, and then there is a mass spectrometry analyzing the content, the microelement, the content of the otolith. Also for this, I mean, the elements that are mostly used is manganese, magnesium, strontium, barium, phosphorus also. There seems to be very nice indicators of the age of the fish. At least they show these seasonal patterns that we can interpret as something that changes with the year. So can be used to help us to age the fish. So nowadays there are many different techniques that you can use when it's not easy anymore to look at the otolith, just looking at the rings on the microscope. Okay, so here I just show an example about the tagging. So they're tagged and released, and you see where they have been moving in that area. So up on the left side, you have the date and the time. So German was the first country, tagging, then Denmark, and then Poland, and then Sweden. So each dot is a fish that's been caught, released, and then caught again after a while. Of course, the problem with this kind of analysis very quickly is just that we have the initial point of fire, not what the fish has been doing during this time, of course. But you can have an idea about, more or less, about how much the fish are moving in this area. And there have been fish out for 4 or 5 years. So there are nice data because we know how much one single fish has been growing during these years. And one nice thing with all these studies about otolith microchemistry is that microchemistry gives us an indication of the environment where the fish has been living. Because for example, strontium is indicating the salinity, phosphorus is indicating the growth rate, for example, and other things. So by using otolith microchemistry, actually we are able to follow the migration pattern of the fish. Okay, so this has been one project that we started in order to answer the question if the growth rate of cotton will change or not. And the answer was yes. That it seems that from the 90s to this last few years, the growth has been decreasing by 100%. Okay, it halved. Okay. From 11 centimeters per year to 4 or 5 centimeters per year. So the growth has gone down. So what can we wait? Okay. So actually the growth has gone down. So the growth has gone down. So the question now is, we want to understand why. Okay. And the last question is, if we understand why, are we able to do something about that? So from the management point of view, can we do something about, in order to get up again the growth rate of cod, or there is nothing to do? I mean, if it's due to increase in temperature, what can we do? Can be one climate change to the fact that humans might carry on there. But is due to something that we can solve. So can we do, if it's due, for example, to the fact that there is not enough food, for example, for cod in the water, can we do something to increase the cod in the water? If it's food.
But they are full of sprouts.
Sorry?
They are full of sprouts.
Full of stuff, yeah. We see. They don't eat only that, we see. Okay, so now this is again the change in the growth rate. So if it is a change in the growth rate, that is a problem. Why the growth rate will change? Now we go back to that. So it seems not to be fishing. Okay, not a problem of fishing. It's probably that each individual animal don't know.
I don't pray. If I need something very energetic in terms of lipids or proteins, I can have a different program per curve.
That was great. Would
As you were saying, maybe the genetic variant, slightly pressure on less performance than
But this even if they're connected to fishery, they select. So they take the fast growing fish, leaving the slower growing fish. So it's not really a decrease in individual growth rate, but it does we have left and see the fish that are growing slowly compared to the other.
also if we have increase of the water temperature, maybe the metabolism of the crops is adapted to all the temperature.
And so we go back to everything we done before, so temperature, oxygen, and
Prey availability and prey type, prey quality.
Five the rates. We are in product composition. The microbial composition of the gas changed based on the stress or environment. Maybe it's not dysbiosis.
Okay. This we did, yeah. We didn't look at. Many of you are studying microbiota, right? You did? Okay. We do with the Candela. Everybody with Candela?
No,
Not you, okay. Okay. But these are, yeah, could be lack of food, but also quality of food. Could be increased competition, for example. Or can be due to physiological stress, temperature, oxygen, and other things.
you said it's incidence of parasites.
Yes, yeah, it goes under physiological stress. So, but yeah, it's coming back anyway, that problem. Okay. Do you think you need to take another break? Or no? Shorter than before. Okay, yeah, well, let's take.
Posso imparare a prima valore senso poi la partita della certa volevamo togliere qua lì non è poi si può una domanda da rispondere e voi studiate le domande. Non è che faccio io che fosse per trovare un modo di aprire esatto Cioè, noi abbiamo detto, chiaro che dentro la nostra quella po', un po', insomma, però facile domande molto semplici. Io sono non vedo. Guarda, vedete, le quattro specie e sono quelle che cambiano da una all'altra. In altre quattro specie non ci sono nell'altra sì, e quindi
Sì,
ci sono, ma non le guardo.
se devi fare un po' di raccontato.
Sì, non le voglio mettere per la adesso diventa insomma
Troppo variabili.
ce l'abbiamo, sarebbe salita, letteratura, qualità, ora che la stata.
Famiglie.
Sarei così, ho scritto fisso che lo ma stiamo facendo.
In questi casi
No, io ho fatto stanziamento con Trophic.
e non è per due secondo me.
Io sono una che lavoro in ispirare la biologia che viene istruita a studiare.
No, noi anche. Quindi assolutamente si deve raccontare perché se mi metto a guardare un variabile
Non ci riesci.
finisci.
Io devo dire, so, non le fanno per parlare. Non c'è nessuno che non questo. Ah, bene.
Se l'ha detto c'era una ricompensa per i pescatori. Ah.
Sì, ma mi va pochissimo, cioè, forse è una cosa.
Noi ci abbiamo provato con le sonde. Non credo.
Che tra l'altro, no, la camminata c'era, ma proprio simbolica.
Cioè, penso che i dottori, perché forse le sonde si staccano di rapido, io penso che sia perché secondo me i pescatori chiamano, no? Per poco, So lì bene, so quello no, perché posso partecipare ad specking e alle punture con questo antibiotico. Sì, sì, aveva fatto tutto perché l'aveva fatto Pierluigi particolare con le razze e noi la volevamo pigliare sulle cordole che avevamo inventato. Ciao, ti posso immaginare, sì, sì, però cioè, dipende se, siccome noi abbiamo durante il sole, tutto gradi, quindi il posto è una maneteria da buona dove noi abbiamo il porto, gli altri leggono il CNR, fanno mo che vogliono questi.
Guarda che guardo se forse c'era scritto sulla
Che noi non avevamo ricompensa, la prima cosa che avevo chiesto mi avevano detto
prima, secondo me la ricompensa, aspetta, forse c'è scritto qua qualcosa, va bene, vediamo. Aspetta, vediamo se c'è scritto. No, qua sulla cartolina non c'è scritto, però sì, c'era una ricompensa, mi ricordo.
giusto per
Quindi giusto per fare il gesto che gli diamo
mi togli una risorsa e te la ripago, cioè quella.
ma magari non so, rimborso spesa se ci spedivano il pesce o di solito andavamo noi a prendere, perché non venivamo in spedizione, andavamo in macchina, andavamo a prendere.
Per tutte cose, per molti pesci, solo che vediamo, se lì non funziona, io non credo che dirà.
Si dipende dalla rapporti che ci sono fra scappato di no, mi ricordo che abbiamo fatto tantissime, poi associato qui, con i paesi, eravamo sì, poi spiegavamo queste carte, però sicuramente tutte le capenze.
Le mostrate, ne hanno fatto tanti sogni, ma non li vogliono. Spiegano.
Stesso che non so.
Però vabbè, io li difendo, sono anche bravi.
Poi tagliare tutto.
Spaghetti, sì, spaghetti e l'antibiotico era quello lì che è blu.
Sì, sì, una cosa
Sì,
voilà.
che fa il vabbè, era una prova, in realtà era tipo una prova per vedere, non c'era dei sole, vediamo se tante soglie che c'era un ragazzo che sta venuto per noi, sto qualsiasi non credo ne so, e voleva appunto vedere come diventare, aveva visto, cioè, quanto crescevano, perché noi andiamo, sì, 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 aveva crescita, quanto tempo era passato e in quanto tempo crescevano, sempre quello.
No, questa diciamo che lo stevo.
Dai,
Stanno lì,
io sempre suo si ipotizza che tipo il settentrionale sono diversi, si incontrano neanche.
Dove ricordare? È una questione di navigare. È importante.
Allora, va bene, dai, continuiamo. Ho avuto una domanda interessante alla vostra collega. Se davamo una ricompensa ai ricercatori, no? Per 240. Sì, pochissimo, è una cosa indicativa. Ah, no, in English, proprio. So, well, give me, I think something like that already. 20 euros, 30 euros was just symbolic, just to give something.
Not for fish.
Sorry?
Not for any fish. Like that, you give 20 euros.
No, perché questo
or a fish, if they could right. 20 euro for 25,000 Sweden. How much is like per kilo?
Okay, but it was very symbolic anyway. But before I forgot to show you something, because here we use this thing or this panel. So it's something that okay, we did to understand the growth of the fish, and can be partially used to look at the migration if you have a lot of data. But of course, if you want to understand better the migration, you need to use another kind of test. And if you want, I show you another experiment that we did, but with the bluefin tuna. Bluefin tuna also. And I think it's quite interesting. So we take a break from cod because otherwise, just to talk about cod is a bit boring, maybe. So some of you maybe already know. So I try to explain a bit the story. So you know that there are two stocks of bluefin tuna. There is the Atlantic Sea. So there is one that is a stock. There are the fish that are spawning in the Mediterranean Sea. And then there is the Western stock. It's the one reproducing in the Atlantic Sea. There are two different stocks. And then it's after production, the needs in the Atlantic, the North Atlantic. And the stock went down very much in the 60s. And the stock back in time was distributed all over the North Atlantic. And it was also present in the Scandinavian first. So you could find tuna out of Iceland, Greenland, out of Norway, Scandinavia, and so on. But then after the stock had collapsed in the border, but then suddenly, 10 years ago, fishermen started to observe bluefin tuna jumping again in the North Sea. So that we wanted to understand that these fish were coming from the Gulf of Mexico, so it was the Gulf of Mexico stock. It wasn't the Mediterranean stock that were coming up in Scandinavian waters. So in this case, we used this kind of tech. These are top-up tanks. So they are tanks very expensive. I think they cost $5,000 each. But we are applied close to the dorsal teeth also for bluefin tuna. But they are nice because they measure parameters in the water. So they are devices that we can measure the pressure, so the depth of the fish. They can measure temperature, and they can measure the light intensity. And the light intensity is used to understand the latitude of the fish, because these are migratory fish, so you want to get tuna, they spawn in the Mediterranean Sea, then they exit to the Mediterranean Sea, and they go to Canada, the USA, and then come back, spawn again. So they move a lot. So you see that red dots, that is close to Scandinavia, on the right side. So we started the project 10 years ago, which we want to understand if the fish that were coming in Scandinavia were reproducing in the Mediterranean Sea or in the Gulf of Mexico. So we were hiring recreational fisheries to catch tuna. And tuna, you know, there are big animals, right? That can be around 2-3 meters long also. So the recreational fishermen are very interested in to participate in this project because they were going out with the fishing rod. They used a herring, like a bite. They were fishing, and we were looking to catch some tuna.
And we were able to tag some, I don't know, 10 to 15 every year. And they are huge animals, so it takes sometimes 4 or 5 hours to pick up each of them by many. And then we were doing very nice way, stuff like that. So actually we were able to tag some and release back and to follow the migration. So now I show you this video. I can enlarge a bit. That is a lecture that I do in natural sciences. So wait, I take away some things that are not maybe important. Okay, so you see this dot, this red dot is one fish that has been tagged in August, because they come up in Scandinavia in August. So they reproduce in Mediterranean Sea in spring, summer. And usually the reproductive areas are, some of you are studying bluefin tuna. No? Yeah.
I fish for it.
You fish, even though. So they are reproducing the area between Sardinia and Balearic Islands in that area. So they reproduce here. And then after reproduction, they go out the Mediterranean Sea and they move. Okay. So here in Scandinavia, they arrive in summer. Okay. Usually they follow the prey. Usually the prey are herring and mackerel, for example, and they follow the migration of this fish. So we caught them in August. And here you see one example of fish caught, released in August. And then you see some plots here. There is a plot about sun, so it's an indication of the latitude of the fish. Then one SST is the temperature of the fish, of the water where the fish has been living. And then BAT is bathymetry, so the depth of the fish, because this tag measures also the pressure. Yeah. So by combining these 3 parameters, you can reconstruct the migration of the fish with some uncertainty around. Okay. So now we start, and you see where this fish has been moving. So wait. Okay. So look, this is the dot. So that is August. And then one important thing is that these are nice tags because you can set them to release, to get detached from the body of the fish when you want, basically. So we set them to release the fish after 12 months because we want a 1-year cycle of the lifespan of the fish. So after 12 months, actually these tags leave the fish. They go to the surface, and from the surface they start to send all the data to a satellite, and the satellite sent to some computer. Okay, because from underwater they can't communicate with the satellite, so they need to get detached from the fish, get to the surface, and send the data to the satellite. So let's see this fish, what it has done. Let's see. Okay, let's do it in another way. Let's do it this way. Maybe it's enough anyway. Okay. So in autumn, this should be winter, more or less. And then when spring is approaching, gets in the Mediterranean Sea, where we know that there is a spawning location. And then after a while, it leaves again, goes up again, somewhere in Scandinavia. And I don't recall if it was caught by fishermen or if just the tag came to the surface. I don't remember now. So you've seen that, okay, in August, so the fish from there went to the North Sea, the Central Atlantic, off the coast of France, Spain, Bay of Biscay, then went out again. And then in spring, probably due to spawning, so to reproduce, goes inside the Mediterranean Sea. So that is a strong indication that is
a fish that is of the eastern stock. Okay, so the stock reproducing in the Mediterranean Sea. So you have seen here this plot. You have seen that as how they changed. So the light intensity, the temperature of the water where the fish was, and the depth. Okay, so by combining these parameters, you can estimate the position of the fish. And you see that the fish is surrounded by two colors, no, one red and one yellow. And these are the confidence intervals, so the uncertainties, how much we are sure that the fish has been actually in that position. Okay. Okay. So we have been tagging not many in some years, and most of them have come in the Mediterranean Sea during spring, but there have been 3 or 4 that actually went to Gulf of Mexico. So some of the fish that we caught here in Scandinavia were from the western stock. So they were going to the Gulf of Mexico to reproduce, and then actually came back. So they can perform very huge migration. So this is just another way to how you can use this tag to, in this case, to understand more the migration. In the other case, instead to understand more the growth of the fish. Okay. Okay, we go back to cod. So we did this experiment. So the growth rate has declined. So now we need to understand why, and you already suggested something. Okay, so let's try to see some figures about this, about the food, about the competition, about the physiological stress to understand more. So this is a plot showing the food intake of cod in time. So on the x-axis you have again time, and on the y-axis here on this plot, on the left plot, you have the energy intake. So the amount of energy that actually the cod was taking from the diet. Because in the Baltic Sea also, again, the Polish, the Lithuanians, the Latvians, they also collected the diet of cod since the 40s, 50s, 60s. So actually using those data, we were able to understand the change in the diet of cod in time. And then applying some formula, we were able to reconstruct the amount of energy that the fish were able to collect, looking at the stomach content, look at what they had in their diet. So you see that this curve is quite similar to the growth curve that we saw before. So it was there's been a peak around 90s, and then a drop, and then from 2000, 2010, also after, the amount of energy that actually cod would take from the diet has been very low. And if you remember, the growth curve is very similar to this. And another thing that we observe is a change in the prey composition, as one of you told before. So not only the amount of energy changed, so the amount of food in the stomach changed, but also the prey composition. So back in time, the diet was mainly constituted by benthic animals. And these, especially this, which is isopod, is an isopod that is called Sadua, the Aintomon, which in the Baltic can reach a size of 7, 8, also 10 centimeters, living on the seafloor. And back in time, the cod was eating a lot these animals. But then recently, in the last 15, 20 years, cod is eating more and more, in proportion at least, pelagic fish. So the small scrap, remember this. So it seems that not only the total amount of the food, but also the prey composition changed a lot in time. And this isopod, I mean, contains a lot of energy. It's quite easy to catch because can move, but also is an animal that can live on the seafloor. So for cod, it's much easier to catch probably an isopod than pelagic fish that is swimming in the pelagic water. So probably also the energy spent by cod in catching this fish has been changed in time. One interesting thing is about the pelagic fish, that the pelagic fish spatial distribution has changed. Time. This is the distribution of the cod. So remember that cod in the beginning was distributed almost everywhere in the Central Baltic Sea. And then with time it got contracted in the southwest, probably because it's the best place to be for cod since it's marine species. But sprat, that is the main pelagic prey for cod, actually changed in the opposite way. So it became more and more concentrated to the south, to the northeast. So to this area outside the Gulf of Riga, the Gulf of Finland, and so on. So it seems that there's been a spatial mismatch between the predator and the prey. So the prey are not anymore in the place where the predator is for different reasons. So that's why the amount of this fish in the cod stomach also decline in time. And other things we think is related to anoxic areas. So with the increase in anoxic areas, or was the reproduction change? I can tell you why in a moment. The distribution of the cod change, because we have seen that when there is no oxygen on the seafloor, what does it do with the cod? Or get pelagic, or you go to the coastal area. So it changes feeding behavior. And probably increase in anoxic areas also caused a decline in the benthic species. So for example, Saduria declined in all this area. And we have seen that Saduria was important prey for cod in the past. We're talking about reproduction. Reproduction is a bit outside the growth rate story, but oxygen is very important for reproduction because the eggs to be fertilized, they need a certain amount of oxygen. And the problem with oxygen is also that since the salinity is very low in the Baltic Sea, if the salinity also declines, what happens with the egg? Remember that the eggs are pelagic, so they float in the water column. If the salinity declines, what do you think they do, the eggs? With a lower density, they sink. They sink, and if they encounter anoxia, they die. Okay. So that's why also if there is anoxia on the seafloor, even if the cod eggs are pelagic, when they sink, they die because there is no oxygen down there. Okay. So that's why also the reproduction declined a lot during these years. This is a situation worldwide of hypoxia and anoxia. That is a paper from 2022. You see that especially in coastal areas has been a lot of problem with anoxia, but also this problem with anoxia start to be relevant also in the open oceans. So there are very big areas now with declining oxygen, which of course can be due to eutrophication in coastal areas, but in the open sea can be due to stratification of the water. So increase in temperature, basically. Okay, because you know that when the water increase in temperature, what happens to the gases? Yeah, they don't get dissolved in the water so well, no? And the oxygen is a gas, no? So if the temperature increase, the amount of oxygen can be dissolved in the water decline. So that's why also the oxygen dissolved from the water decline with increase in temperature. So it's a global problem. Okay, in the Baltic Sea, what happened with the cod? So if we had before a community where the cod actually is a top predator, but is feeding mainly in the benthic habitat. So especially is this Saduria, so this isopod, but also some pelagic fish. With no oxygen on the seafloor, what happens? The cod become pelagic and proportionally start to eat more pelagic fish. So probably a prey that it doesn't want to eat so much because much more difficult to catch and it spend much more energy to catch. Okay, so that is the situation of the Baltic Sea nowadays with what they are called dead zones, so the areas that are totally dead, and all the life is in the surface. So what is the link between anoxic areas and cod growth? Okay, the link can be many. So one is about the compression of the distribution of cod. So if the anoxic areas increase, there is less habitat, of course, for the cod. So the habitat declines. This can provoke more competition between the individuals because they are concentrated in a smaller area. There is a direct physiological stress because if the individuals live in low oxygen areas, they need to spend more energy just to survive. And there is an effect on the benthic food. So without oxygen, of course, the benthic food also decline. Another thing that has been observed in an experiment is that the fish eat less when they are exposed to hypoxia. So if they are in an environment with low oxygen concentration, they lose appetite. They're not hungry anymore. They don't eat. And this is something that seems to be related very much to what we see, that if the cod stays in a habitat with not anoxia, but hypoxia, with low oxygen, that they don't want to eat. And that's what we have observed also in the stomachs, so that the stomach content is much lower now than back in time. So they don't have appetite, probably because of this. Okay, this is another explanation. So we have seen the oxygen. We have seen that the change in spatial distribution of the prey. And then there has been another hypothesis that is the one of selective fishing. Remember that I told you that since until 2015, there was this minimum landing size. So it means that the fishermen would land only cod bigger than 38 centimeters. Okay. So it means that everything below 35 centimeters couldn't be caught, basically, because it was not possible to sell. So some people were suggesting that actually this caused an increase in competition of the small fish because they were not able to be caught by the fishermen. An increase in the density of the small fish, this was initiating density-dependent effects, so that the small fish were not able to grow to big fish. And this, that's why one of these people were suggesting that actually to decrease this competition within the small fish population, we should allow the fishermen to fish also small fish, so that to thin down also the small fish in order for the ones that were left to grow quicker. Okay. Another problem is the seals. We have already said this before. The fact that 50, 60, 70% of all the cod that we catch are full of these parasites. Of course, when you see a fish with this kind of liver, of course, you can imagine that also is not very in a good state. Okay, so this can affect a lot also the growth rate of cod and also the survival of the cod. So another suggestion that has been put forward is to increase actually the hunting of the seals. And that is something that is already happening, especially in Finland. They catch seals. Okay, and in the last years, the quotas of the seals has increased in order to try to save the cod. Okay, these are all management decisions that the politician ministries are taking because in order to try to save the cod population. And this is the figure showing how the seals is the final host of these parasites. Okay, so these are all the possible reasons for the fact that we observe a decline in the growth of cod. So we said oxygen, hypoxic areas, the change in prey distribution, the change in the diet of the cod, maybe temperature, also the parasites due to seals, and so on. So now the question is it possible to get back to the previous situation? So is it possible to get back to the situation where the cod population is good, in good state? So many fish and big fish, or no? So in the case that we can do something about that. Okay. So for what we have said, what do you think? Do you think it's easy to get back to the previous situation? So of course, it is a choice, right? So for us, is it better to have a system, a community, an ecosystem with a lot of code? Or is okay as it is now? So it's okay to have an ecosystem without code, I would say. Because many times also a political decision is okay, this environment or not? Do we want to get back to the old environment or not? And if we want, is it possible to do it?
It's to be embarrassed. You can go back somehow. There is conservative agriculture case, but it is expensive and very effort, and it needs a lot of effort. So everything has to be on and for energy.
Yeah. Okay, but if we wanted to get back to the previous situation, do you think is easy from what we have said or not?
The directly on the fisherman now, you can put a stop or an invitation for fisheries in the area restoration. But I think the other problems like the environment, temperature, salinity, oxygen, really difficult to change it.
Because all the other measures, like the cod fish has been closed, as I said, for 8 years almost now. But there is no sign of recovery. But it's something that we observed also in other stocks. For example, the cod stock in Canada. I don't know if you have heard about the stock in Newfoundland. There was a huge stock also there in of cod. And in the 90s, also more or less, it crashed and never came back. Even if the fisherman was banned, the fishermen started to fish on other species, but still the cod population didn't come back. So it seems that when a stock is overexploited, so it has been overexploited for many, many years, and it crashed, it's very difficult to get it back. Because in the meantime, the ecosystem has changed because of the absence of the big predator. So that for the big predator is very much difficult to get back. That is a sign of, maybe in ecology, you have been studying this regime shifts, stable states, ecological stable states. That means that when the state of the ecosystem changes too much, very difficult to get back to the original state because in the meantime, everything has changed. So the conditions for cod are not good enough now to get the cod back. So I would like now that we could try to understand why the new conditions are so bad now that for the cod, it's very difficult to get back. If you think about temperature, of course, I mean, temperature increase, so is more difficult. But try to think about, okay, since the cod has disappeared from the system, what has changed in the system due to the absence of cod?
Biodiversity and abundance of species change.
Well, do you remember what changed the most? Okay.
Trees and the reason that they go.
Exactly. So, I think that cod disappears. This disappearance has caused an increase in the pelagic fish. So why do you think this situation now is hindering the cod to come back?
So that was like eating both that microorganisms, and they are not prey anymore of cod, so they are destroying everything.
Destroying what do they do?
The struts are eating what you call it what waste, right?
See, I don't know if an example was waste, but what you say is correct. So if there is so much sprat now in the system, what do? They eat a lot of zooplankton. And cod needs zooplankton. Yes. Okay. So this can be a new system for cod where there is a lot of spray that in theory should be good for cod. But there are a lot of pelagic fish now eating zooplankton. So there is not enough zooplankton, for example, for the cod larvae, for example. So that can be an example of a positive feedback loop. I don't know if you haven't studied in ecology. So that the new system is so resilient that is much more difficult now to get back to the original state. So it can be an example of alternative stable state. So now I show you, this is
Make fish oil and fish meal.
good. We have been thinking about that. So these are all theories that we jump because it's boring. So this is all the feedback loop, the positive feedback loop, which can hinder code to come back. This is just theory. I don't know. I'm not saying that it is working this way. So let's say that we don't have any more cod in the system. So we have a lot of pelagic fish, sprat, shown as the size of the sprat. If there is a lot of pelagic fish, what happens? Pelagic fish eat zooplankton, remember trophic cascades. If they eat a lot of zooplankton, there is not zooplankton left for small cod, right? So that can be one of these feedback loops. The second feedback loop is the one caused by the fact that sprat is eating cod egg. So there is a lot of sprat population, there is a lot of predation of cod egg. The third loop could be the one due to reproduction. So if there is a lot of sprat, there is density dependence in the sprat population. So also the individual sprat don't grow so much because there are too many. So the energy of each fish is not as high as before. So when cod is eating a pelagic prey, this pelagic prey contains less energy than before. And this can explain the fact that the energy intake in the cod diet has declined in time. So actually, since the cod disappeared, his habitat, his environment has changed due to his disappearance. And the new system hindered the cod from coming back. So that it was called in ecology alternative stable state. So it's a new state that is very resilient. So it's very much difficult to break it and go back to the original state. Of course, this is just theory now, but we know that these three mechanisms are occurring. So we know the sprat is eating zooplankton. We know that cod larvae also don't have enough food. We know the sprat is eating cod eggs. Many times the problem is to quantify this effect, because we know that this link exists, but many times we don't know how strong it is. Okay, this is how those feedback loops. And then there is one fourth feedback loop that is more ecosystem feedback loop. So remember what happens when cod disappears. Phytoplankton blooms increase also, at least it's the risk of having phytoplankton bloom increase. If we have phytoplankton bloom, this increases also the risk of having anoxia. Okay, anoxia, right? If we have anoxia, that is bad for cod, right? Because it affects reproduction, growth, behavior, and so on. So if we have anoxia, it's very difficult to get back the cod. Okay, so that is a feedback loop that is auto-maintaining itself. Okay. And then there is another loop that when we have anoxia that is mainly a problem of the deep layers, so close to the seafloor. What happened to the nutrients? I mean, some of you have probably studied chemistry and I don't know, biochemistry. They stay on the seafloor, But when there is anoxia, what happens? That is the nutrients that are in the sediment, they get suspended again. The biochemical reaction. So if there is no oxygen, all the nutrients that are in the seafloor, they get back to cycle, to the pelagic water. And if the nutrients come up again, these provoke even more phytoplankton blooms, more anoxia, and so on. So it's another feedback loop that is auto-maintaining the situation, the bad situation that we have now. Bad for cod, at least. Okay, so if we go to option for management, okay, what can we do for cod? So some of you already told something that can be, I don't know, science fiction or no, but but it's true. Can be an idea. Can be an idea. So what would you do to save the cod? So let's say that we want the cod back. And maybe we don't want it. But why we want the cod back? Because there's a lot of culture behind. I mean, people in Scandinavia, they want to eat cod. I don't know why. It's not even tasty, but they want to eat cod. Now there are a lot of flatfish, for example, in the bones. And they are also good fish. So we can ask ourselves why we want this cod back, actually. Let's say that we want the cod back. What can we do? Sorry? I don't know what, but yeah, one of the most commercial important ones, yes. But in this area, in the Baltic Sea, I mean, it's very at the end of its distribution. So here is more popular, maybe herring is more popular, but also cod is very important. So actually, these people, these countries bordering the Baltic Sea, many of them, they don't want to start to eat flatfish. They want to continue to eat what they are used to eat. So herring, cod. So they want to get back this cod. And many times I say, but let it go. So it doesn't matter. Eat something else. I mean, but they want the cod back. So what can we do to get it back?
And another species between cod and stratus. That can be a prey of the cod and a predator of
I think, I mean, it can be seals. I mean, seals eat everything. So they eat both cod and sprat. So that is a bit difficult to understand.
all the time. To find some species that cod is species.
Now, between cod and sprat, there is nothing, let's say. So one idea, you had one idea. What?
We farm cod.
Okay. No, in nature, we want to find it. No, but you had a good idea. So one idea that we had, or the European Commission had in the past, was to fish more on sprat, actually. So fish more on sprat in order to release these positive feedbacks in order to have a higher recruitment production of cod. That was actually an idea, to fish more on sprat. And this area, sprat is fished not for human consumption, because it's a small fish, quite thin, so it's not for human consumption. But it's fished for oil, flour, and fish food. Aquaculture, I mean, for aquaculture. But especially for oil and flour. Was some
It was we found this because we found the mechanism where the broken mechanism, if we try to change another thing, risk discovering another mechanism, what we just stand in the way.
Okay. So let it alone. Yeah.
Yes, it's easier enough. Here it's not that big.
But one solution that some years ago was suggested was to fish more on spread. Another solution is that we can do something about seals. So there are already some countries that are hunting more seals in order to release a bit of this problem about parasites, liver parasites, especially the Finnish are doing. Another thing that we didn't speak about is about flatfish. Actually, cod is a big predator also of flatfish. So especially when the cod was big, like in the 80s, 90s, it was eating a lot of flatfish. Now, of course, there is not big cod anymore. So this predation pressure on flatfish is not existing anymore. So the flatfish also has increased. And probably now the flatfish population compete with cod for this aduria, for this benthic prey. So another suggestion that somebody has put forward is to fish more flatfish, which is also good because instead of eating cod, you can eat flatfish if you want fish on the table. So this is always a political decision. Okay, we want to continue to eat fish or not? Do we want to eat the local fish, or do we want to import fish from Canada or from Mediterranean, or we want local fish? So another idea would be to eat more flatfish. Okay. And eat less cod, and probably also this will decrease the competition between flatfish and cod. That is another one. So I can show you. So there is every year I told you that there is this stock assessment. So it's all this calculation trying to understand the status of the different stocks. This is a procedure that usually is followed in the European Commission. So you have your sampling at the national level. I mean, everybody working in CNR here already know this, but this biological sample, and then all these data are put all together at international level. And when we talk about the Baltic Sea, is ICES, which is that symbol, which means International Council for the Exploration of the Sea. So putting together all the data that each country collects. And then every year in Copenhagen, there are people estimating, the abundance of the different stocks and to understanding the status you good or not. And then with this data, ICES give management advice to the European Commission to say, okay, you should decrease the fisheries, or you can increase even the fisheries depending on what objective you have, and so on. And then the ministries, so the European ministries decide something, and these will cascade down on the management decision. So there are fishermen that have their annual quotas, they need to use some kind of gears, they need to avoid certain areas, certain season, and so on. So that is a cycle that is going on every year. Okay. So the suggestion in the last years have been, you remember that cod and sprat back in time, they were overlapping in space. But then from the 90s onward, the cod has been moved to southwest and the sprat to the northeast. So actually one suggestion that it was in one of these, in this meeting was actually not to fish sprat in the southwest, to leave more pelagic food for cod. So basically we were saying to the fishermen, okay, don't go to the southwest to fish sprat, go to the northeast where there is a lot of pelagic fish, so that there is more food for cod in the southwest. Of course, this is very, very difficult to do because if you are a fishing boat from Estonia or Finland, it's good to go to fish on the northeast. If you come from Germany or from Poland, they prefer to fish much closer than to go too far away. So of course, what we do every year is to try to give biological advice, so advice based on biology of the fish. But then the politician need to think about economy, about social problems, about many other things, not only about the biology of the fish. Another suggestion has been many years ago, instead, to fish more sprat, as you say, to release these positive feedbacks and maybe to have a higher reproduction of cod. Another one is to fish more on flounder. Here you see the relationship between cod and flounder that are eating both the same isopod. So fishing more on flounder can mean to have more benthos for cod. So increase the growth, increase the reproduction, increase the population. Another one is to hunt more on seals. If we believe that the link between parasite infestation and cod growth is the one that is driving the decline in cod growth. So these are all different management options that actually have been proposed during the years. None of these have been applied ever. And what it has been applied so far has been the closure of the cod fishery. But as I said, we didn't see any recovery of the fish, which is typical of the population that has been overexploited for many years. Then it's very difficult to come back. When the population goes below a certain value, then even if you stop fishing, it's very difficult to get it back because the ecosystem has changed because of the disappearance of the predator. So it's very difficult. And so actually this is the end of the story. I mean, that is a very sad story. And because this Baltic Sea is one of the areas that is most studied in the world, but still people were not good enough to save this population. Of course, as I said, I mean, the cod is marine species. In the Baltic Sea is at the limit of its distribution, so it's very sensitive. I mean, so many times when I speak about this thing, I say there is no future for cod in the Baltic Sea because in the future temperature will increase, salinity will decline, oxygen will not improve. So there is no space for cod in this area. So it's much better to focus on something else. This is my point of view. Let's see something else. No, just some few words, then we finish and we can speak about what you want. So the Baltic is a very sensitive sea. We said that it's closed. It's brackish. Brackish means the salinity is very low. Low biodiversity. There are many forces acting in the Baltic Sea. There are 9 bordering countries, one is Russia, so the political situation is not very easy for the management also point of view. Cod is in a bad situation, what is called distressed situation. And the main problem is that the population not only has declined, but also the individual fish has been smaller and smaller in time. We think to know partially why, so the declining growth rate, even if we didn't understand perfectly why the growth rate has declined. And one of the key messages that many times we think that we know everything already, we know all the biology of the fishes or the ecology, blah, blah. But for example, this is the case in which we still don't know many things about the growth, for example, of the cod. So one main point is that basic, ground science is still very much needed. Okay. And one last point is that cod has been at the center of the public debate for many, many years in the Baltic Sea, but there are many other species that we shouldn't forget that play a role in the lack of recovery of the cod population, but those can be very important species for the commercial use instead of cod. This is, we have been talking mainly about this species here, but then there are a lot of also seabirds that was also an impact on the fish, cormorants, guillemots, so on. There are seals, there are flatfish that can be used much more than cod, for example, for human consumption now. there is these new fish, sticklebacks. In Italian, I don't know if it's come? L'halibut, no, è un pesciolino. No, lo stickleback is. No, I don't remember now the Italian name. That is very small fish with spines, and it has increased exponentially in the boat in the last 20 years. And then now the fishermen want to start to fish on this, not for human consumption because it's, there is nothing to eat, but for oil, flour, and other things. And so the Baltic Sea, that is considered also very low biodiversity system. Okay, there are many other species to study. Come? Lo spinadello, okay. Okay, so that was all. Do you have a question? Some questions?
I think yours, Russia is definitely for the fishing position.
No, that's a big closed in Europe to catfish. So I just told me that already, so no serious. So they continue, but I think that their impact is not very good.
That's update in 6 years.
So you can read in 6 years if you want to.
I'm shut now of the shades, but
That is what Seth told me, and then I didn't really have a story for it, but I decided to figure out the Seth story. No, because there are not biological interesting aspects that really try to what can be, reproduction. The fact that they start to reproduce when they are very small, when they are 20 centimeters long, they start to reproduce. There are always signs that the something is. Okay, then good luck and
For me, would be
what was up? The actual of today you something going?
I thought for the email italiani, there are some guidelines for the exam,
Yes.
and usually it's
Psychology patterns. I know.
no, because usually some teachers show during the lesson the type of essay or testing, and also the teacher. You really talk about the some. If not, we can send it by email and via. The deadline is in June, so
One more kind of example.
yeah.
What kind of personal
Everything up to you, so the question will cross depend. They have to do 10 exams. There are 10 exams in June, so yeah, we try to balance the type of stuff.
I will let you know.
Yeah,
So what can you?
thank you.
Okay, thank you. Fine.
Aspetta. Vediamo tutto.
Okay.